So today we're going to learn a little bit more about mutations and how those can affect your genes and eventually your traits. So today what we're going to do is we're going to focus on kind of 3 key points, and then we're going to go over some examples of mutations. Just briefly remind you about kind of what mutations are. We're going to talk about what causes mutations, and then what sorts of effects they can have. So let's just remember that mutations are a change in the order of bases in the genes, right? So you have these A's, T's, C's, and G's. A mutation is just when we change your genetic code, and then that results in changes in your proteins. We've already talked a lot about different types of mutations. We talked about chromosome mutations, where you might change the shape of a chromosome, or we talked about genome mutations, where you may lose or gain a chromosome.
I remember that from yesterday.
That was yesterday, exactly. Today, after we go over a little bit more causes and effects, we're going to talk about single gene mutations, where we have very small changes in your DNA that only affect a single gene. Ooh. So these might affect things like your hair color, your eye color, but not necessarily have these detrimental effects on your entire genome. So let's just quickly go over a couple other things. What causes these mutations? And we already talked about a few of these causes. We have 3 causes. Number 1 is we can have mistakes in DNA replication when DNA copies itself. We could have mistakes in meiosis when we make our sperm and eggs. Or we could have different mutagens, chemicals, or different physical environmental things like radiation that causes our DNA to mutate. I'm going to talk about each one of these in just a little bit of detail here. So mistakes in replication can happen. So when we go and copy our DNA to put it inside a new cell, we can accidentally make mistakes. It's very rare. The statistic they throw out is 1 base in a billion gets replicated wrong. And then even then, that one single base has to be a base that affects a protein to actually have any sort of effect. And these errors commonly get picked up. We talked about an enzyme called polymerase that builds the new polymers of DNA. They can actually proofread. So they're able to see when some bases aren't pairing up and go back and fix that. Because remember, you can't pair up a G with a C. It just doesn't work that way. So anytime one of those bases doesn't pair up, it recognizes that they're not fitting together and that it can go back and go ahead and repair that. The other one we talked about, and we talked about this in depth yesterday, was mistakes in meiosis. So remember, you're supposed to separate your chromosomes by half in order to make your sperm and eggs. But occasionally those pairs of chromosomes won't line up, and when that happens, you end up with sperm or eggs that have too many or too few chromosomes, right? So you can see over here, maybe these are sperm cells. These guys got paired up correctly. These guys didn't split, so this sperm has too many, one too few. So those can cause mutations as well. And then the big one, the one that we kind of focus on today, is mutagens. So mutagens are any sort of, well, I guess they can be physical, chemical, or biological factors.
You guess?
Yeah, well, it's a pretty good guess. It's there, right on there on the screen.
Didn't you make this?
Once upon a time.
All right.
Any of those factors that can kind of damage your DNA. Big one is radiation from the sun. We all know that tanning too much can cause cancer. So that's because that radiation actually damages your DNA. Other things like different radio waves and X-rays can destroy your DNA. And then we have chemicals. So there's chemicals, a group called base analogs. So this is pretty crazy. We know that DNA has these 4 bases, A's, T's, C's, and G's. Well, some chemicals are similar enough to these A's, T's, C's, and G's that they can take their place. So they can actually damage your DNA this way.
What's an example of one of those?
I don't know any. I haven't consumed any chemicals underneath the sink, but don't do that. And then there's different agents or chemicals that can change the structure of the DNA, just damage the DNA. And then biological, even some viruses can infect hosts by inserting their DNA into them, and that can actually infect your DNA and cause mutations. A great example is the HPV virus is known to cause cancer. That's a big topic right now in the news. Yeah. And then let's just quickly talk about the effects of mutations. The effects of mutations can actually come in 3 forms, and this makes sense. You can either have good mutations, bad mutations, or neutral mutations. So the first thing is we've talked about mutations so far as being a bad thing, but there's certain examples where it can be a good thing.
Evolution.
Evolution, and a great example of this is HIV resistance in humans. There's actually a group of people, so the HIV virus you can see here attaches to these proteins on the outside of your cell. Well, there's a few people in the world who actually have a mutation in their DNA that causes them not to make this protein on the outside of their cells. Because they don't have it, the HIV virus cannot actually attach to their cells.
So humanity is saved.
So they are immune to the HIV virus. So there's actually people who can't get AIDS. It's pretty crazy.
Where do they live?
A lot of them in Europe, actually.
That's not helpful.
Yeah, not to you anyway. An example of maybe a bad mutation, which we've talked about some examples already, but certain deformities like this frog that has an extra leg, that's probably bad because it's probably bad because he probably can't walk right unless it makes him walk faster and stronger, then it's a good mutation, but I'm assuming that this is a bad mutation in this case. And then we could have neutral mutations that have absolutely no effect on us. They may make us look different, like for example, blue eyes is a mutation in your genes that gives you that eye color.
That would have been really good in the 40s.
But, what?
That would have been good in the 40s.
But that mutation doesn't have any negative effects. It doesn't really have any positive effects. It doesn't really do anything for you. So, yeah.
Except save your life in the 40s in Europe.
Okay, so it's not the 40s. And then just a reminder that these mutations ultimately affect your proteins because DNA gets changed through your RNA, through your proteins, and that's what's affecting our traits. So what we're going to talk about now is that example of mutations, those point mutations I talked about, those single base mutations. And these are pretty interesting because these are the mutations that we can model and we can actually see their effects kind of right away. So we've already talked about these mutations a lot. We can skip through this. So what we're going to talk about is actually these mutations that affect just a single base, but that ultimately affects a specific amino acid and then changes your proteins. So let's take a look at this. Point mutations or single gene mutations that affect a single DNA base. A single DNA base. And they come in two categories. So the first category are called substitution mutations, of which there's 3 of. And the second category is called insertion and deletions. And these cause something called frameshifts. So I'm going to focus on these substitution mutations the first. So a substitution mutation is exactly what it sounds like. It's when you substitute one DNA base letter for a different DNA base letter.
Like math.
Basically, yeah, you're just changing one letter to a different letter. And it comes in, like I said, 3 forms. So the first form is called a silent mutation. And a silent mutation is when there's a change in the base letters, but the new amino acid or the new codon still codes for the same amino acid. That is, the new amino acid is the same as the old one. Let me give you an example of this, and I've done this in RNA. CAC could be a 3-letter codon. Let's say that last letter, that C, mutates to a U. Right? So we have a change in a single base letter. Well, if we look at our wheel, let's figure out what both of these make. So CAC, C-A-C, gives us histidine.
Oh, sorry.
Now let's see what the mutated version, CAU, gives us. C-A-U.
Histidine!
Also gives us histidine. So both code for the amino acid histidine.
Right,
You saw
I didn't see the answer below.
Nothing changes. So in this case, we had a mutation. There was a change in the base letter. A C changed to a U, but that caused absolutely nothing to happen to our protein. We call it a silent mutation because we cannot see its effects. Let's look at another example. This is called a missense mutation. And this is when there's a change in the base letters that changes an amino acid to another amino acid. So let's take a look at this. We might have a codon, GAU. Let's say that A mutates to a C over here. So now we have GCU. Again, that's a 1-letter switch, but let's see what the effect is. So GAU, if we look on our wheel, GAU becomes aspartic acid. We change that to GCU, 1 letter, now we have alanine. So this is what we would think of as a mutation. One amino acid changes to a different amino acid. We call that a missense mutation. There's one more type of mutation called a nonsense, and this happens when there's a change in the base letters that changes an amino acid to a stop codon. So again, we're changing the letters, but this time we're not getting a different amino acid out. We're getting to one of the 3 stop codons. So for example, if we had a base of CAA, let's say that mutates to UAA. So again, we're only changing one letter. We're substituting one letter for another. But let's look at what that does on our wheel. So CAA gives us glutamine. And then let's look at UAA. UAA brings us to a stop codon. So in this case, one amino acid became a stop. We call that a nonsense mutation.
So that changes the most.
Well, this, you could think of it as if you're building this protein, normally this protein, let's say, is supposed to be so forth and so long, right? Well, now you're building this protein, and then all of a sudden it stops.
So does it continue to build another protein?
No, it doesn't, because if you remember from a previous lecture, you need to have that start codon to tell where the protein starts.
Where's that on the wheel?
It's AUG, methanonine.
That's very specific.
Yes, all.
That's the TEM I couldn't find yesterday.
Yeah, well, it's met. It's just we squeezed it in there. But yeah, okay. So it has to start with that methanonine. So unless this is a methanonine maybe right here, you're not going to be building another protein. So this,
that really screws you up.
That really screws you up. And generally what will happen is this protein that you now make won't be functional. It won't work. It won't do anything. So again, that can either be a good thing or a bad thing, depending on what that protein does in your body. If it gives you HIV resistance, it could be a great thing. But in this case, 9 times out of 10, it's a bad thing. Yeah, you probably need every protein that your body makes. So this might be a practice problem that I give you, and I want you to be very strategic about these practice problems.
That exact problem, those letters too?
No, I'll switch up the letters.
Sorry, kids.
Generally what I'll do is I'll give you kind of a sequence of DNA, or in this case I've just put the RNA sequence. Let me put this in parentheses so everyone knows. This is not a DNA sequence. Sometimes I'll give you a DNA sequence, but in this case I've given you an RNA sequence, and then I've given you the original and the newly mutated sequence.
Oh, I found it!
And I want you to figure out what type of mutation it is. Now, before you get excited, wait. Before you get excited.
Yes, Mr. Massey.
Right?
Sorry, Mr.
What I don't want you to do, Mr. Howarth, is I don't want you to translate this entire sequence using the wheel.
Just look for the change in the letters.
Just look for the change.
I see it.
Wait, remember, let me show you the right way to do it, Mr. Howarth, that we read codons 3 letters at a time. So what I want you to do is
Starting from the left.
Starting from the left, yep. So you're going to just block off every 3 letters.
Those are the same.
Yep, those are the same.
Those are the same.
There it is, Mr. Howarth. There you go.
I found it.
So once you find that mutation right there in those 3 bases, now you can go ahead and figure out what type of mutation it is. To figure that out, we'll have to figure out how it changed. So let's go ahead and use our wheel on the original sequence right here on CCC. What does CCC give us?
Proline.
Proline.
Oh, it begins with a P.
There you go. Let's see what it turns into now. Now we have UCC. UCC. Serine.
How do you remember these?
Years of biology. So if we change proline to serine, what type of mutation is that?
I wasn't taking notes, but substitution.
Well, they're all substitution. So substitution means we change a letter, which we did. So you're right, it's a substitution mutation.
100.
But they're all substitution mutations.
Right, well, it's not. It's not a no sense or nonsense or whatever, because that's where it ends.
So you have 2 other options.
It's not the silent one. Crap, what's the other one? It's a non-silent one.
Silent?
No, it's not silent, because silent doesn't matter. It would be
Nonsense.
Nonsense! No, wait, what's the other?
Missense.
Oh, shit. Missense! Sorry, I swore in your video.
A missense. A missense mutation is when we change one amino acid to another. And if you forgot, Mr. Howarth, you can always just go back and look through this video at the 3 examples.
I'm pretty sure if I messed your slideshow up right now, you get mad at me.
Okay, so one last type of mutation that we're going to talk about, and this is called a frameshift mutation. This one's interesting. It changes the reading frame, and it changes every codon in the sequence. So what would cause that? Well, there's 2 things. The first thing is an insertion. So far we've talked about substitutions where we change one letter to another. An insertion happens when we add an additional base. So we just sneak another one in there. So for example, GATTACA might turn into GATATACA. So if you notice, what did I insert there?
An A between the T's.
Right, I inserted an A between the T's. So that's an insertion mutation. I add a letter.
Is the other one a delete? Oh, damn it.
The other one's deletion.
Deletion!
A deletion occurs when you remove a base in the sequence. So I could have G-A-T-T-A-C-A. Maybe I just remove something at the end, a C-A or whatever.
The A. You removed the A.
I removed the A and I put 3
You
lines.
removed the A, but...
But, I deleted some letters, right? You get the idea. We removed some letters from the sequence. But both of these, insertion deletions, cause what we call a frame shift. That means that, and this is going to sound confusing at first, the reading frame is changed. So every codon after the addition or deletion is then changed. So let me give you an example here. Let's say we had a sentence, the fat cat ate the rat. Now, first of all,
They're all 3 letters.
They're all 3 letters, just like our DNA codons. We read 3 letters at a time. So let's say we delete the first letter. Let's say we delete this T here, but we still read the codons 3 letters at a time. We always read DNA 3 letters at a time. Everything is going to shift over 1, and we get hif, ada, right? Because all the letters shifted over 1. We still have fat cat ate the rat, but now we still read 3 letters at a time, and everyone bumps over 1 space. So that changed everything, right? Let's say if we inserted a letter, let's say after the word cat, we just add an extra T. So we still have the fat cat ate the rat, but now it reads the fat cat tata ether tit. So by adding or taking away a letter, we screw up everything after that, because everything has to shift over 1 space.
You took some random languages in college, huh?
Yes.
You read that pretty efficiently.
So you could see for an example here, right, we might have our codons here. Let's say I add a C, I sneak a C in there. You can see here AUG, CCC, these are regular codons, and then maybe I put AUC, and then what happens? Well, now I'll read GCC, CGA, CUA. And you can imagine, I don't even have to go through it right now, but if I were to use my wheel and look at the original AUG, CCC, and now I look at what I get, AUC, GCC, CGA, these are going to give me entirely different amino acids on my wheel. So everything after that insertion is going to be messed up or made a mistake, right? So that
Are we done?
We're done.
That's an extra bonus slide, kids.
Well,
You draw a happy face there?
Yep.
Do you need help drawing a happy face? That's a horrible happy face.
He's giving you a thumbs up.
He's giving you the middle finger.
He's giving you a thumbs up
He's on a fishing hook.
because you've done a
How many are? Oh no, wait.